So the first session for the day will be Introduction to Biosimilars by Dr. Pawan Kumar Singh from Biocon, Bangalore. This session will be chaired by Dr. Melvin George. Dr. Melvin George, he is the professor of Department of Pharmacology from SRM Medical College Hospital and Research Center. He is a consultant of clinical research at Hindu Mission Hospital. He has pursued his MBBS, MD Pharmacology and DM Pharmacology from JIPMA. He has published 75 scientific articles in international and national peer-reviewed journals, and he is also the author of 8 chapters in textbook. He is an investigator in several multicentric clinical trials of various diseases like heart failure, acute coronary syndrome, stroke, dyslipidemia, and chronic stable angina. He is a member of IEC at Global Hospital and Hindu Mission Hospital, and he is an organizing secretary for this workshop. The next chairperson for the session is Dr. Kala. Dr. Kala is the Associate Professor from Department of Pharmacology, SRM Medical College. Madam has done her MBBS, MD Pharmacology and Diploma in Clinical Pharmacology and Postgraduate Diploma in Geriatric Medicine. She has a clinical experience of over 25 years and teaching and research experience for 9 years. She was awarded gold medal for securing university rank in MD Pharmacology. She is currently the medical scientist for Institutional Ethics Committee at Sri Sai Ram Ayurveda Medical College Research Center at Tambaram. Ma'am, kindly introduce the speaker for the session, ma'am.
Thank you, Dr. Indumati. Am I audible?
Yes, ma'am.
Yeah. So good morning to one and all present here. A highly experienced clinical pharmacologist with considerable national and international experience with the pharmaceutical environment is none other than Dr. Pawan Singh, Senior Medical Director, Biocon Biologics, Bangalore. I am deeply honored and privileged to introduce the speaker, Dr. Pawan Singh. Dr. Pawan Singh had completed his undergraduate from GS Medical College, Mumbai, and MD Pharmacology, DM Clinical Pharmacology from PGI Chandigarh, the Institute of National Importance. Sir has started his career as Manager Medical Affairs at Dr. Reddy's Laboratory, Hyderabad. Later, he had climbed the ladder in various companies such as Pfizer, Serano Clean Farm, Sun Pharma Advanced Research Limited, Glenmark Pharmaceuticals Limited. Now, Sir is in Biologic, Biocon Biologics at Bangalore as Senior Medical Director, Clinical Development. To his credit, he has a number of publications in peer-reviewed national and international journals. He has got vast therapeutic experience in the first 3 phases of clinical pharmacology and the area of pain, diabetes, and oncology. He is having the interest in the area of clinical pharmacology and clinical development in diabetes, pain, immuno-oncology, and biosimilar. Sir is going to enlighten the topic biosimilar. Sir, we are eagerly waiting to listen your talk, sir. Over to you, sir.
Yeah, so before Dr. Pawan begins, I this time want to say something about this topic. Is going to be the Dr. Pawan. I just have to thank him, especially because he has been the scientific chairperson in the sense that he's been helping us in formulating this whole program. We have sat together and discussed many things over how best we could do this program, and he's given a lot of time for that. I wish to specifically thank Dr. Pawan for that. And coming to the session, this will obviously be the session that will set the ball rolling for the day. It will help us understand how biosimilars as such is different from the development of biosimilars, how it is different from the development of generics, how biosimilar, the regulations that are there with biosimilars are progressing over the years, and especially when it comes to the challenges faced with biosimilar development, being in its structure, being in the analytical data. So some basic overview we would be able to understand from this session, after which the subsequent speakers will take each of these issues in a much deeper way. So thank you once again, Dr. Pawan. Over to you. You can share your screen, Dr. Pawan.
Hey guys.
Dr. Pawan, are you there?
So, Dr. Melvin and others, can you hear me?
Yes, we can hear you.
I'm sorry, using the Bluetooth and it's giving some issues. No
Yeah,
problem.
we can hear you now.
Yeah, I am sharing my screen and then probably will start.
Yes, you can share.
Okay. My screen visible to all of you?
Yeah, it's visible. You could just put it in full screen, sir.
Yeah, sure.
Yes, it's fine.
Okay.
Go ahead, sir.
So before starting, very thank to the Dr. Melvin and Dr. Kala for the nice introduction. The words are too much for me. I am just a small person who just try to help Melvin in organizing. The basic aim is that new generations who are coming and might be coming in this field should know what they are going to have. And if interested, they can pursue their rest of the career in this area. And this is going to be very exciting area, to be very honest. And the topic which is decided by Dr. Melvin for me is Introduction to Biosimilar. So it will be a gist of all the topics which we are going to cover in the throughout the day in more details, but I will just be touching and going to most of the topics. And the aim of my talk will be just to give some terminologies and more from the regulatory perspective, because everything related to biosimilar changes and will keep on changing as the regulatory scenario will evolve. And that is the reason we have kept also two topics on the regulatory scenario, one international and one also for the Indian. So my name is Dr. Pawan. I am working as a senior medical director here at Biocon Biologics and working for the development from phase 1 to phase 3. So ideally in biosimilars, there is no phase 2. Mostly we work for PK/PD study as phase 1 and then phase 3. And also developing the strategy, interacting with various regulatory agencies and to harmonize the clinical development plan, which fulfills the need of most of the regulatory agencies. Just a disclaimer that whatever I am going to speak in this workshop is my own thoughts and should not be attributed to the organization with which I am affiliated, that is currently Biocon Biologics. So the first and foremost problem when we talk about biosimilar is what is the terminology to be used? Because if you see across geographies, the regulatory guidance have different nomenclature. The most common term which is used in day-to-day is biosimilar. Similar biologics is also used mostly in the EMA and India region, and follow-on biologics is the terminology given by FDA. If if you have to give a one mark answer to this question, what is biosimilar without explanation, you can say it's only a generic equivalent of biologics. But this is not that straightforward, which we will come in later part of my. So biosimilar are basically a similar biological product, and they are only similar in terms of all these three words are very important: quality, safety, and efficacy, in reference to an approved reference biological product, and that similarity is based on comparability. We will see more about what is the comparability of what we are talking here. Now, if you talk about the legal definition based on the US, this comes from the PHS Act and followed by PPACA healthcare reform. And it defines biologic as any virus, therapeutic serum, toxin, antitoxin, vaccine, blood component or derivative, allergen product, protein, or analogous product, which are applicable to the prevention, treatment, or cure of a disease or condition of human being. So in a very simplistic way, we can define biologic as any product whose active ingredient is made in a living system. Now in US, with reference to this, there are three words which we should focus: biologic, biosimilar, and interchangeable biologic. Now, this interchangeable biologic is the concept which is currently only applicable for the US but in most of the other regions, we have only two options, that is Biologic and Biosimilar. So as I told, Biologic is a simple and practical definition is that a product in which the active ingredient is made in a living system. Biosimilar is again a biologic product which is approved based on showing that it's highly similar to an already approved biological product, and that is it has no clinically meaningful difference in terms of safety and effectiveness from the reference product. I will touch some part on the clinically meaningful difference also, because that is a word which has got immense implications while we develop our studies. And third is interchangeable biologics. So FDA designation that switching patients between such products can be made at a pharmacist level, no intervention of a healthcare provider is required, is interchangeable. And Dr. Viraj will be talking more about this topic about interchangeable biologics. So in many ways, biosimilars are to biologics the same as generics are to the drugs. However, the name does not change and the scientific and regulatory principles are established for all biologics. Now, if you see biosimilar, the presumed problem which we see is that it is about the establishment of similarity, while the actual problem is not the similarity establishment, but its origin, which is from a living system or from a bio. So this similarity can be solved by comparability, which established the highly similar analytical standard as a way to define a biologic. But the problem, as I said, is more about because of its nature of bio and its production in a living organism. If you compare this small molecule generic versus biosimilar, the major difference is that small molecule are low molecular weight and low complex molecule, while biosimilar are very high molecular weight and have complex 3D structures. Generic require chemical synthesis, while biosimilar are produced in living organization. And manufacturing process for a generic is easy to reverse engineer, but for biosimilar, it cannot be replicated as such. And the active ingredients are actually identical copy in case of generic. But in case of biosimilar, although they're required to contain the same primary amino acid sequence, biosimilar is not identical to the original, but highly similar to that. So as we discuss that generic drug, which is defined as FDNCA Act, is equivalent, while manufacturing changes to drugs and biologics, you have to establish the comparability. And then based on totality of the evidence, you have to prove that your drug is not clinically meaningful difference compared to the original product for biosimilar. But in a nutshell, identical science applies to all these applications, and semantics do not change the underlying regulatory science, and quality is the foundational for all medicines in all leading markets. Now, with respect to biosimilar, if we see because these are more of a protein in nature, it can be compared with a key where the non-operative part of the molecule is allowed to have some differences in composition and conformation, but the biologically active part of the molecule must have the same composition and conformation. So non-operative changes may occur in all biologics, not only biosimilars, based on manufacturing conditions. This slide basically gives a pictorial representation of the complexity of what we are talking with generic versus biologic and biosimilar. If you see aspirin, it is about 21 atoms. Ramipril, for example, is having around 62 atoms. Insulin, again, is around 790. But what we are currently focusing and developing is monoclonal antibodies, where the molecular weight ranges up to 20,000 atoms. So because of the sheer size of the biologics or the mAb, their manufacturing itself is very complex, and you cannot have an identical or equivalent, but you can only develop a highly similar product to the reference biological product. However, once you get the approved, this complexity is no longer a relevant argument. Now, coming to this definition of similarity. So, based on different definitions, actually there are three different determinants in the definition of biosimilar. The first is that obviously it should be a biological product. Second is the reference product should be an already licensed and approved biological product. And you need to demonstrate high similarity in the terms of safety, quality, and efficacy, which is a must. So at these three levels, quality, non-clinical, and clinical levels, you have to demonstrate the similarity in comparison to the reference biological product. And the development process is much more rigorous compared to generics, because for small molecule or generics, you only need to have a proof of quality and bioequivalence, which is normally done in healthy volunteer, and you can show the equivalence. And you don't require a very substantial clinical data, and references, you can refer to the originator's data with respect to other package. But in case of biologics and biosimilars, because there are different manufacturing process, and they often yield differences in the end product which is there. And after the quality of a biological medicine is demonstrated, there are still some non-clinical and clinical studies which are necessary. And because of there can be changes in the manufacturing processes, and there can be some impurities or difference in the tertiary structure. Immunogenic response cannot be predicted, and therefore it has to be tested and proven that it is not higher compared to the originator product. So this is the pyramid which shows how we try to establish the similarity in compared to the reference biological product. So the sponsor must first show that the candidate is highly similar to the reference originator at the analytical level. And this can take multiple iterations in early stage development before clinical testing may begin. And the sponsor is also required to perform detailed analysis of the originator reference, especially if the structure and biological function is inadequately defined in public domain. So we need to do the physiochemical characterization followed by biological characterization, then non-clinical study followed by phase 1 PKPD, and then phase 3 clinical trials. So the whole, if you see, the base of this pyramid is analytical. So the whole focus of a biosimilar development, though from a clinical perspective, we don't go into the detail of that, but our R&D colleagues are doing a wonderful job in establishing this analytical similarity, because this is the base of this pyramid. If you are not able to show the analytical similarity at the CMC level, there is no way you can move forward and develop a biosimilar product. And this analytical similarity requires a very highly extensive structural and functional characterization. And as part of our basic, we are not into that domain that much, but I will touch some part of it, that how extensive this analytical similarity program is run. Now, we need to first understand the molecule per se, and this requires almost more than 45 different methods which provide information on multiple attributes, what we call as orthogonality. And each and every attribute is evaluated more than once for the redundancy. So if you see, you need to evaluate primary structure, impurities, biological activity, then post-translational modification and higher order structure. And combination of these attributes by some algorithm, AI and machine learning is also coming into place to understand this. So it requires an integration of data from multiple analytical and biological tests, which provides complete understanding of the molecule. Now, this part I basically try to cover what are the differences in the generic versus biologics and biosimilar, and what are the basis of establishing the biosimilar, and some part on the analytical similarity part. Now, why we are focusing and talking about biosimilars so much? Obviously, as one way we are, as discussed and introduced, that these biologics are a kind of game changer in treatment and management of various diseases, but because of the high cost, they are very prohibitory. So what are the implications of introduction of biosimilar in the healthcare system, and what are the opportunities we have? I'm going to touch in the few subsequent slides. So if you see the commercial impact of biologics, almost all the blockbuster drugs which are recently developed and came are biologics. And if you see their sales, it's crossing millions and millions of dollars. So these are the commercially most impactful products, biologics, compared to the small molecules. And now most of these molecules in the coming decade are going to lose the patent, and biosimilar is expected that they will come into the market. So there is a huge commercial impact of biologics and followed by biosimilar. Now, this development approach of biosimilars, when we talk, is very close to originator compared to the generics. So this slide actually talks just that comparative, that a generic requires around 2 to 3 years to come to the market compared to biosimilar, which requires 7 to 8 years. And clinical studies which are required for generic is just be studied in healthy volunteer, but biosimilar typically phase 1 PK/PD study and a phase 3 comparative safety and efficacy studies required. Even the phase 3 trials in a biosimilar is quite huge, more closer to a NCE development. And typical sample size is 500, and sometimes depending on the margin, it may range up to 2,000 also. And post-approval activities for a biosimilar is also more closer to a new biologic compared to a generic. And Dr. Mudgal will be talking more about phase 4 and pharmacovigilance of biosimilar. With respect to opportunities, again, till 2019 itself, 21 important biologics have loose patent protection. And biologics represent a very cost-effective opportunity for patients, mainly for MABs in area of oncology, autoimmune disease, and cardiovascular diseases. And the total market value of biologics is more than $50 billion currently, and more than 150 marketed biologic products are worldwide, with almost 500 products are still under development. And the market has itself grown to 6.22 billion by 2020. And currently, almost India is part of all biosimilar development plan due to more clarity on regulatory guidelines in India. And Dr. Maulik will be touching more about the regulatory guidance which are currently applicable in India and how can we leverage India for development of global biosimilar product. This slide you need not to go through, but this only depicts that what are the major area where biologics and biosimilars are working. And as you can see, it is mainly about the inflammatory disease and oncology, and some in the cardiovascular metabolic disorder, and few in infectious disease. But with this COVID coming, you have seen that many biologics are coming in this infectious disease part also. Now, why one wants to invest in biologic therapeutics R&D? So basically, in terms of scientific and clinical, it required specificity, multifunctionality, and because of very prolonged half-life of this biologics and durable efficacy and safety that they provide compared to the small molecules. In the terms of commercial, 10 of 20 top-selling drugs are biologics, and this number is going to increase, and biologics are going to take more share in the terms of top-selling drugs. And 8 of 10 best-selling biologics are monoclonal antibodies, and antibody-based drugs contribute around $75 billion of biologic therapeutic sales. And in the terms of regulatory, 17% of first-in-human antibodies lead to regulatory approval. So the passing percentage, the rule of 10%, if you see, it's better in the terms of biologics compared to small molecules. And as we know, we are all part of mostly the clinical development part. So in the terms of clinical, we have to do PK, which required comparative pharmacokinetic studies, appropriate design. It can mostly single dose, but sometimes we need a study dose and multiple dose study. Then in the terms of PD, comparative PD study, and we need to select the most relevant population. It's not that always we need to do it in Hindi. We can go for patients, especially in the patients in the area of oncology. And now the most of the regulators, especially the European Union or EMA, is focusing more on early identification of PD markers which are clinically relevant. And their expectation is that if there is any clinically relevant PD marker, it should be included in your phase 1 PK/PD comparability study. In terms of safety and efficacy study, comparative safety and efficacy in relevant patient population and nature, severity, and frequency of adverse event has to be monitored. A safety and immunogenicity study, so you need to have the pre-approval and post-approval assessment of the safety and comparable safety data with adequate patient exposure. And especially though not required by the EMA, but FDA definitely want at least one transition period to know what happens after switching from reference to the biosimilar product in the terms of immunogenicity and hypersensitivity. And then extrapolation to other indications can be done. It depends on similarity on the quality, preclinical results, then safety and efficacy result, and based on mechanism of action, which are similar or different with respect to the approved indications. This is an interesting article which one can go through, which talks about the end of phase 3 clinical trials in biosimilar development. And I hope Dr. Shrikant will be touching more on this with respect to UK, which is coming, not currently in the EMA or FDA domain. And if that comes true, then probably we can get the first biosimilar approval in UK without doing any phase 3 clinical trial. Hopefully, and let's see, because as I said, the whole basis of establishing biosimilarity is more dependent on the analytical and CMC similarity. And phase 1 PK/PD study, phase 3 contributes, but there are too many variables in establishing the biosimilarity in terms of phase 3. Whether a phase 3 waiver can be granted or not that depends on your similarity in the terms of analytical similarity and your PK/PD study. I'm not going into the depth of this. We'll leave for the Dr. Gattu to touch on this. But only thing is that if this goes, then the job opportunities which we have in pharmaceutical industry for clinical development will become redundant. And at least I don't want this to happen till I got retired on a lighter note. So no one should require a scientifically invalid and therefore unethical studies. So if a biosimilarity can be proven without phase 3, definitely it will reduce the timeline for approval, reduce the cost of development, and ultimately it will translate into lower cost of biosimilar and more access to the patient population who actually needs these therapies. Now, at the end, the biosimilar development requires an optimal integrated strategy at all the levels, whether it is regulatory, clinical, or commercial. Regulatory, again, as I said, will be test-based. Clinical, Dr. Sudeep Sinha is going to take after my lecture about the challenges what we face in the clinical development. And commercial part, again, Dr. Dang will take in the terms of real-world evidence required, improvement, and other studies which can help decision making by the payers. So in the terms of strategy to succeed in the biosimilar development is to choose wisely in which area you want to venture, which map you want to select depending on the patent expiry, depending on the capacity of your company. Then unlock the assets, and it's very important to partner locally in the terms of local payers, the local CROs, and the regulatory bodies. So to summarize, biologics can be thoroughly analyzed and characterized, and biosimilars are systematically developed to be highly similar to their reference biologic. The clinical studies are mainly aimed to confirm the characterization work. Extrapolation builds on the entire similarity exercise, and post-authorization studies continue to monitor safety. Biosimilars must meet the same quality standard as originator products, and biosimilars may increase patient access to biologic medicines and contribute to savings for healthcare system. So this is, I think, in a very brief, I have talked about biosimilars, mainly the introduction of some terminologies, the basis of proving biosimilarity, and our subsequent speakers will talk more in the depth of challenges in the clinical development, then interchangeability studies, followed by regulatory and the post-marketing safety and pharmacovigilance, and real world evidence, which is now coming as a large thing, especially in the developed world. Because only proving safety and efficacy is not going to help. You need to show the benefit, and also the decision maker are not only the regulatory bodies, the insurance providers and the payers are now having a much bigger say in making the decision for availability of any biosimilar in their respective market. So with this, I would like to thank again the organizing committee of this workshop, and thank you, Dr. Melvin and the entire team, for giving me the opportunity to interact with all of you. I hope I have justified with the time and not taking too much time of all of you, and we'll be happy to answer questions, because it will be better to have more interactive session and to answer your queries rather than just talking one way. So with this, I would like to end my talk here. Thanks. Over to you.
Yeah, thank you, Dr. Pawan, for that wonderful introduction to biosimilars, how biosimilars are developed, how they're different from the way generics and the way biologics also as such are developed. So I just had one question before which I'll take up, and Dr. Kala also will ask questions
Yes,
on
sir.
this topic. So the first question that I had is, sir, you said about CMC, about this with respect to CMC and analytical information. What is the status in India? How many pharmaceutical companies are really having the tools available, resources available with them to get into this? Because I believe this is a very complex area. What exactly is the current status on this?
Sure, Dr. Melvin. And to be honest, a very wonderful question, because when we are looking, we don't go into the depth of CMC, but while working, we face that this is the real challenge in the development of biosimilar. With respect to the companies which are currently working in this area of biosimilar, one is my company, Biocon Biologics. The other important organizations are Dr. Reddy's Lab, which are doing good in biologics. Lupin is working in this area, and Zydus, which Dr. Malik will be sharing more thoughts on that. And also Sun Pharma is working in the biosimilar. So though they have not developed any biosimilar which is in the public domain, but in the terms of biologics, they are working on the Tildra, which they have got the approval also. So they have that manufacturing capacity in the terms of biologics and biosimilar. Apart from that, I don't see there are too many players in this area which have this capacity or this kind of setup to develop biosimilars.
Okay. Thank you, Dr. Pawan. Dr. Kala, you can probably ask a few questions. You're on mute, Dr. Kala. Please unmute yourself.
Yeah. So, thank you, Dr. Pawan Kumar Singh, sir. So, such a nice explanation about the biosimilars. I think everyone got a clear idea about the biosimilars. So, even I have some doubts, sir.
Sure.
Yes, sir. So getting approval for the normal drug from the clinical drug development and getting approval is a lengthy and definitely it's a toughest process, sir. So is it the same they are continuing for the biosimilars, giving the approval and the process is the same or a little bit reduced?
So compared to original biologics, little bit reduced, but not very much reduced. The reason being, you still have to do a phase 3 trial with the biosimilar, which is the most time-consuming part even for the novel biologics. We don't need to do a phase 2 study, but phase 1 and phase 3 is required. And with respect to R&D or CMC, it actually requires more time compared to the original biologics. So overall, there is a reduction of only couple of years compared to the original biologics, not very much.
Okay, so then the biosimilars we have in the form of monoclonal antibodies for the autoimmune disorders and the anti-cancer drug. So the cost of the original drug is a little bit high, definitely. You do agree with that. Then what's the cost of the biosimilars, sir?
Okay, so as I said, I will give a comparative. For example, if a generic molecule, so if the originator is costing around 10 rupees, a generic can be available in 1 to 2 rupees. But in that comparison, if the original biologic is around 10 rupees, you cannot expect a biosimilar to be less than around 6 rupees. So there is a reduction of around 30 to 40%, but not in line with or in the scale of generic, because biosimilar development also requires a very extensive capital expenditure. So there is some reduction, but not too great compared to generic.
Yes, that's also a very great opportunity for the medical fraternity, sir. Thank you, sir. Thank you very much for your wonderful talk.
Okay.
Definitely, ma'am. Thanks.
Thank you, Dr. Kala. So I'll just take a few questions from the delegates. So there's one question. Do phase 1 studies in biosimilars focus on safety or efficacy or both? From
No.
Dr. Sujay. Yes.
Phase 1 study neither focus on safety nor focus on efficacy. The primary objective of phase 1 is only to establish a pharmacokinetic equivalence. So you do like generic, you also do a phase 1 study, PK study for the biosimilar, and you have to establish that it should be falling within the equivalence criteria of 80 to 125 predefined limit. Sometimes the margin can be tighter depending on the molecule, but that has to be predefined. Safety as a part is evaluation, but that is not the major objective. So the major objective is to establish the PK equivalence, and now, as I said, EMA expectation is also to have a PD equivalence in the phase 1 as a secondary endpoint, not as a primary endpoint.
Okay. So there was another question whether biosimilars are being developed because biologics are expensive and to maintain the patency period. So I think that you have already answered in your talk. And whether biosimilars can be used for minor ailments, or is it something only for chronic diseases or cancer? So are we even looking at minor ailments? That's what.
No, so because of the cost itself, you don't want to go for minor ailments treatment with this costly. When you have a more efficacious and safe options, only where there is an unmet medical need, where we should venture with these costlier options in the terms of biologics and biosimilars. And for biosimilars, you will not get any new indication. It is always what is already approved for a biologics.
Is there any dose difference with biosimilars?
No. The dose has to be same as of biologics. Otherwise, you are not a biosimilar.
Yes. And considering the complexity and cost of development of biosimilars, are the cost savings really significant, both from the company and patient's perspective?
So as the same answer I have given to Dr. Kala, there is a significant difference, but it depends on from where you are comparing. If you are comparing with generic versus the originator, that is not that much. But if you compare as such, a 30 to 40% reduction in the cost is, I believe, is a significant reduction in cost. But again, it varies from molecule to molecule, cannot be generalized. But this is the overall ballpark reduction in the cost.
Yeah. What is the reason for not having phase 2 trials?
Okay. Now the objective, if you see it for the phase 2 trial, is on mainly identification of the dose which you are going to use for a phase 3 study. So recommended dose for phase 2, and also to have a early proof of concept of efficacy. So in a biosimilar development, these objectives are not there. The only objective is that you have to prove that there is no clinically meaningful difference from the originator. You cannot use a different dose also. So there is no need of doing a phase 2 because it did not contribute anything in terms of establishing of similarity compared to originator.
Thank you so much, Dr. Pawan. I think we have answered all the questions in the chat box. So with this, Dr. Kala, you have any other questions?
No, sir. No questions, sir.
Okay. Thank you.